All right, we are rolling. Jamie Andrews, welcome to the Terrain Theory Podcast.
Hi Ben, how you doing?
Very well, thank you. We're excited to have you on, and I thought we could just start with some background, so the audience knows who it's dealing with here.
Yeah, I my name's Jamie Andrews. I moved to France about 6 years ago, just over 6 years ago, out into the wild, the open space into southwest France, looking to move out of the rat race in southeast England, not too far from London. I saw what was kind of happening politically over there with Brexit and things like that. And I saw things kind of closing in and where it was leading to. So I decided to move out, buy some land, and I bought a Maison de Maître, which is a bit like a chateau, a small version of a chateau. And I am doing it up into an off-grid house. So I have all the electricity is run by solar panels. I collect rainwater off the roof into large vats and run a small holding and completely heat the house with wood pellets that I make through sustainable means of finding sawdust at local sawmills and heating my house for free with it. So trying to pave the way, moving away from, the government and that kind of system, but hopefully in a not too, kind of how people see, moving off grid as, growing a big beard and moving into, a forest and speaking to birds are your only friends or whatever, trying to do it with style and show that it can be a positive maneuver and something for the future. So, yeah, I have a little bit of scientific background. I studied geology at university in the UK at a reputable university, but I made up my mind when I was there that, and I started to see a lot of what was going on and glimpses into what would be the should we call it the profession of science. I saw that higher academia started to actually not expand my mind. It was telling me, and I use this phrase a lot, and I was lucky enough to have some very good teachers at school who taught me how to think and not what to think. Yeah, and they're two polar opposites. Is one teaches you, how to discern your environment, how to discern what is in front of you and make your own decisions. And the other tells you what to think and tells you what to do. It's a system of hierarchy and a system of, top down, if you like. And I saw in my geology degree that was a very blunted process where they started to dictate what science was, what you should think, how things should be done. And yeah, that's how I kind of I left left university there. I completed my degree. I got a degree in geology, which has stead me nothing. It's I have found it of no use whatsoever. Went and did my own thing. I'm in the property business now, so that's what I do for my income. But yeah, and this kind of led me up to 2020, this massive gulf within science that I didn't use, and then when 2020 came along, I started to, and I had no idea about virology at all. I didn't even know that the premise that viruses didn't exist and contagion wasn't a thing and terrain theory. I may have heard about Antoine Béchamp, but the very deep recesses of my mind. And so what really kind of sparked me off was just the data that was coming out. 2020 came along. It seemed very, the what was being told to me didn't match up with what I was seeing with my eyes. And when the data started coming out, I was looking at death statistics. They were saying, you got those rolling feeds on CNN or whatever, and they said this many people have died here, and this many people have died here, and I have quite a few connections into India. My wife is of Indian heritage, of mixed kind of Indian heritage. And so a lot of her family are over in India. She has connections into Mumbai and New Delhi. And in 2020, in kind of February and March, we were saying this thing kind of took off in China. And yet in India, they were saying one or two, a few people had this supposed disease in these major cities. There were, handfuls of people. Yet their death statistics stayed almost, rock bottom for about 6 months. And it didn't make any sense to me statistically. You have people on the streets, New Delhi and India are known for their quote unquote unsanitary conditions with people living on the streets. And I was sat there just scratching my head, contagion wise. You have everybody living, tooth to cheek right next to each other. And supposedly this thing wasn't ripping through the population. And so that really aroused my suspicion, through March, into the summer months. And I started just to look at kind of causation. I just opened Google Scholar, and at the time, I didn't know what I was looking for at all. It was just kind of like giving people viruses. That's what I was typing in. Have you ever given somebody a virus, like on purpose? And I kind of narrowed it down and then started finding some of these papers which were coming forward, and found out they were called human controlled infection models. And they do give people what they consider to be viruses, in titers. And in the 20th and 19th century, they just put people that were showing symptoms of the disease and put them in close proximity, or took their mucus, or took their bronchoalveolar lavage and rubbed it into people, and rubbed it into their eyes, or squirted it up their nose, or injected the blood from infected people into other people. And what was stunning to me was that nobody ever got sick. It was like, this moment in my life, which was just like, hang on a minute. We've been told for all of this time that you can cough at people and you can do all these things. And it was just a light bulb moment for me. And I went and I found every single controlled human infection model ever published. It's nearly 100 papers, and it does it to over 100,000 people, over the course of 120 or 130 years. And this involves everything from all of the scary ones, HIV and polio and smallpox, and even, some very, very old documented ones with the plague. Right the way through to the common cold, where they set up, common cold units and so on and so forth. And I mean, this for me was enough science to say and convince me within my own mind that the contagion of biological pathogens is a myth. And so I live my life by that, and it's extremely liberating, and it's extremely, once you learn that your neighbor can't kill you with a misplaced sneeze, it's just, it's a very kind of liberating experience. And that led me to, actually looking at some of the proof for the existence of viruses. Not that I think that it matters too much, because these, contagion studies prove that even if they do exist, you can't catch it. You can't transmit from person to person disease. It doesn't work like that.
Yeah,
but I
well,
thought
just
that
to,
it
and
was
just to step in real quickly, it's really the 1-2 punch against virology. It's the 1-2 punch knockout punches against virology. One is the discovery of an examination of these contagion studies, is what I've heard them called. Yours is a much more precise language, but Daniel Roydus, who we had on, who wrote Can You Catch a Cold, has done a wonderful job compiling what I imagine is a lot of these same studies that you unearthed in the back of his book. And it's another place where I would suggest to the audience member who is hearing this for the first time, go check out that book, go find some of these contagion studies yourself and realize that it's simply never been proven. In fact, at this point, they've maybe disproven it. So that's punch number one, contagion studies. They've never shown contagion exists. And then two, which is sort of the next step that you're about to launch into, is okay, there seems to be proof within virology that they've isolated a virus, demonstrated that it exists. However, continue.
And that's it. I mean, spoiler alert, because I was going to recommend the book as on the reading list. It's a fantastic book, and you should get it. And it's very very well documented. Basically, I put it up on my Twitter, and it's a bit kind of janky to read, but that does really well in terms of presenting these studies in a good light. And yeah, I think also the contagion studies is something that's very pertinent, is the fact that it shows that nothing is contagious, because obviously we also talk about bacteria and parasites as being part of that, because bacteria objectively exist. You can see them mostly with a light microscope, and so you can define them, you can purify them, you can have what is a bacteria in your hand. It's just that they don't cause disease. And so these contagion studies show that. They've done it with typhoid, for instance. They've tried to give people, typhoid, where they concentrate the bacteria and give it to them, and basically nobody gets dysentery, nobody gets diarrhea, nobody gets vomits in the entire thing. So it's just like it's categoric proof right there. So and this is what kind of led me to finding out about Dr. Lanka's work. He's really an extension of what is the Perth group, who was doing this with HIV, led by a lady called Eleni Papadopoulos Eliopoulos. I always butcher her name. She's definitely Greek, but who has passed on now. But she really did a lot of the control studies, which is what has led me up to what I've been doing. Back in the late 80s and 90s, she did a seminal paper in 1997. Looking at proteomics of HIV and showing that in uninfected cultures there were the same protein bands in uninfected as infected cultures. And she inspired Stefan Lanka, the German. He was a virologist. He is a virologist studying bacteriophages, and is published in studying oceanographic. I don't know. Ocean. Something you find in the sea. Some bacteriophages you find in the sea. He's published lots of papers doing that, and he was pivotal during the fake pandemic. In raising awareness about the fact that science within the realms of virology is anything but settled, as they keep calling it. And he conducted these control experiments, and unfortunately he kind of released them a bit on the kind of on the down low. And I think he was getting to the end of his career and didn't kind of want to make too much of a stink about it. He had already been to court, where he issued a prize money to show proof of existence of the measles virus, which turned out to be a very long and drawn out process in court, which he eventually won. And they had to admit, and I have to get my nomenclature right here by saying they had to admit that they could not find in a single piece of paper scientific evidence that the measles virus existed. And those are the words of the higher court of Stuttgart through a long, drawn-out process of litigation. But he did win, and he did get them to admit that in not a single scientific paper on its own there is proof of the existence. So, and kind of as part of that, he did these control experiments where he looked at the isolation process of viruses. And it's that hook word, the hook phrase that you hear when people say, the virus has never been isolated. And I think a lot of the project that I'm running here is to really explain what that means, because I don't think that people understand. A lot of people do, but I think that, in general, people could kind of say it and not really know how that's come about. And so what Stefan Lanka did and what we've done is take the control, the cell culture isolation, where they take a cell line. So they take cells from, there's numerous, but and they're quite, I mean, they're not particularly nice. They take them from, say, embryonic kidney cells. They take them from the kidney cells of dogs. They take them from fetuses. They take them from all sorts of, they take them from lung carcinomas. They take them from liver carcinomas of dead and dying people. So they take these very fragile and broken and already diseased cell lines. All of them are not from, Arnold Schwarzenegger's bicep. They're from, literally from the lung tumor of a 58-year-old bloke in Japan that's died from lung cancer. And they take these cell lines and they grow them. They use fetal bovine serum. So that's the fluid from around the heart of a young cow. And this fluid provides nutrients to these cells so they can effectively grow. And that's what they do. They put them in a Petri dish and they grow these cell lines out to the edge of the dish. Not too much, because you can kind of crowd them out and they kind of start to starve themselves. They then add antibiotics, and the antibiotics are supposedly to clear from other pathogens. So any bacterial or fungi that may be in the air, spores and things like that. Now, obviously, they shouldn't be there, because all of these things are conducted under controlled scientific procedures. So they're all conducted under a flow hood, for instance, which have HEPA filters, which adequately take the air away from the from the experiment that you're conducting. So they shouldn't do anyway, but they add antibiotics just to make sure. The problem is that these antibiotics are known to be toxic to cells, especially kidney cells, and they use kidney cells a lot of the time in a lot of the cell culture isolation. So what they're claiming is that they're taking these cell lines and they're adding a sample of sputum or mucus or bronchoalveolar lavage from the chest, and when they are seeing they're then removing the nutrients. So they've used the nutrients to grow these cells, and then they're removing the nutrients for whatever reason. I'm not entirely sure. But they remove the nutrients and add a sample of something which they claim contain a pathogen. And then when they see that these cells in the Petri dish are dying, they point at them and go, that's because of what we've put in, or what is in the sample. There's a pathogen that is killing the cells. Now, what Stefan Lanka did was he showed that if you don't add any sample, if you take nothing, you just add the ingredients of the cell culture, and when you remove the nutrients, these cell lines die. They die anyway. So it can't possibly be the addition of anything within the sample that they're adding causing the effect that they're claiming denotes a virus or denotes a pathogen in the sample. Therefore, you've falsified the experiment. Therefore, you've said that, essentially a control has failed. So when you have a control, and I've just released a Substack on this, talking about controls, and I think that they are science. I think that a control experiment is the actual mechanism for science. Science is two things. It's observing what's occurring, and this happens in an area called epidemiology, which any scientist that is intellectually honest, which is a rarity these days, would say that epidemiology is not a science. It's just looking over there and saying, oh, there's 2 diseased people over there, there's 3 diseased people over there, and then they assume the cause. Okay, they say this pattern of disease is because of a transmittable virus, but they never actually go and prove it. The second part to what is science is experimentation, to show the cause. And so without that second part, you're just making up, stories for why these things are occurring. And the control is the mechanism for finding out. And if I can just, because I think it's quite important for people to understand actually the power of control, the power of a control. So if I take you through like what is a very, school type experiment. So if you look at you hear correlation versus causation all the time. So somebody sees people on a beach in the summer getting sunburnt, and they see people selling ice creams, okay? And they hypothesize that ice cream sales are causing sunburn, okay? So we all know that that's not correct, but you can do a valid scientific experiment to show that that's not correct. And that happens with a control. So the control is you take the independent variable, which is ice cream sales or ice creams, and you remove that from a controlled environment. So you take the same variables that are occurring, so a beach in the summer, in the sun, and you remove the independent variable. And lo and behold, people still get sunburn. So what you've proved is that ice creams don't cause sunburn. I mean, it's very simple, but the power of it is phenomenal. It's a safety mechanism, and the safety mechanism. And what I found within virology is that this control, they say that they do mock controls in these things. So you'll see when you read any isolation of virus papers, you'll see them say mock control, we've done the control. They don't write a methodology for it. They never write a methodology for it. They never take you through the most important part of the scientific experiment, the control, to show proof that their cause and effect is what's occurring, is caused by the thing that they're putting in. And so, and I've interviewed a lot of people online, seasoned virologists who admit that their controls basically are not controls. They admit that they're doing things like they'll PCR test it, or they'll keep it in 10% fetal bovine serum, which isn't a control. They just call it a mock control and go, yeah, it's fine, just out of the way. And, and so this is what I'm seeking with the project to do, is to raise awareness of this. But there's a wider push that, and we can go through. We'll go through the presentation of the actual results that I found with this project. I've been working for the past 8 months with 3 labs in the US, with numerous seasoned microbiologists and biochemists, and been commissioned commissioning CROs, which are independent research labs, to do the work for us. It's obviously not me. I'm just managing this project. So we are using the scientific knowledge and scientific processes of the industry, crowdsourcing and crowdfunding this to do science. And here's an important point that I want to get across with with what this project is and what people can kind of bring to the table with it is that we are releasing the methodology, okay, for this project. We're going so far above and beyond what is a virological paper, a normal virological paper, because there are calls, saying the sample is contaminated, for instance. Now, in a standard virological paper, it will never tell you that they've done it in a flow hood. It'll never. You just have to assume that they've had what's called chain of custody, where somebody hands it to somebody else in a recorded and safe manner, and they know that they haven't opened the bottle and so on and so forth. They just assume that all of these processes are legit when they're actually probably not. But what we're doing is going through every single stage. We'll give you the HEPA filter in the flow hood. We'll give you, the cubic feet of air moved in the flow hood. Yeah, we have taken video. Okay, we've taken video of it occurring, and we're releasing the video of the scientist conducting the experiment. And we are releasing that into the public realm. Okay. And the point to this project is not to get this published. It's not to convince any single scientist. I don't care. What this is much, much greater than that. In fact, if somebody wanted to publish this in whatever journal, Nature or so, I would tell them no thanks, because actually, I don't want it associated with that. Actually, I want to steel man this process to give to the most critical audience, and that is the people. That is who counts. And this is the scientific project that I want to push, and what I'm kind of coining as Science 2.0, to push into the realm, crowd-funded, crowd-sourced science, where people can actively engage in a live experiment. Okay, and that's why we have released these preliminary results into the public, so that they can understand the process, they can get involved, they can fund it, they can back it, they can make suggestions, they can change it. Because there's such a gulf between what's going on in science in a lot of areas, but I want to attack science, in this realm. In the way that 2020 occurred, where you just saw top-down orders barked from Mr. Fauci, follow the science trademark, and everybody kind of goes like, in order. It's a hierarchy. And how do you undo a hierarchy? Well, you have to empower from bottom up, and that's what I'm trying to do, is set up other people doing these experiments. And this is the other thing, is that I'm just a layman. I came off of Twitter. Yeah, I opened a Twitter account and started doing this, and this is it. Anybody can do what I've done. You call up and you pay research organizations to carry out the experiments. That's it. It's very easy, and it's just bottom up, And the way that I describe it is, Albert Bourla, who's the CEO of Pfizer, he's a vet. He's a trained veterinarian. He's got nothing to do with the science. He's actually just a businessman, right? He just employs the scientists to do their work. That's all he's doing. And he's got a monopoly, and he's got a stranglehold on what the science is. But he can't compete with crowdsourced type. if it was actually a genuine movement of people wanting to find out the truth, and that's why I'm doing this, really, is just to know for myself. That's how I started out, just going, I actually just genuinely want to see, whether I'm completely mental or not. Is it completely mental to think that viruses don't exist? And lo and behold, some of the stuff that I'll show you later on is stunning to me, because when it came to Lanka's work, obviously we had a fair idea of what was going on with the cell culture. We knew, and there's numerous papers written that tell you that when you starve a cell line, it dies. I mean, it's logical, right? When you take the nutrients away, it's like stopping eating. That's it. You die. But we also conducted TEM, and that's really the crux of this project so far and where we've got to, and that very visceral thing of them pointing and going, that round thing is a virus. And as I can take you guys through the slides that we have,
Yeah,
I'll show you.
let's jump into that for a second. I do want to just clarify a couple things. Clarify, sort of double click on a couple things, as Mark Grober might say, and then give Mike a chance to jump in here real quick. But you mentioned EM, that's the electron microscopy. This is another strategy or tactic that virologists use to quote unquote prove the existence of a virus. This is point and declare. They take that sample, that cell culture sample that they claim contains the virus because it broke down the cell, and then they prepare that sample to view it under electron microscope, and in that process find an image, they point at it and say, haha, there's the virus. So you're going to walk us through what happens in the control experiment, and then when you prepare that sample in the same way to look at electron microscopy. I want to jump back real quick to the cell culture, and again remind the audience that this is really this is sort of the gold standard of virology, of cell culture of virus isolation. This is how they prove, quote unquote, that a virus exists. And it's interesting to me that they use the antibiotics and the antifungals to remove potential variables when in fact they're just introducing another variable.
That's right. Yeah.
I find that astounding. And then
No, it is. When you actually understand it, it's witchcraft. It's it literally has that much in common with just putting it into a witch's brew. It's really quite stupid. Yeah, I just to firm up what you're saying, it's the only way. It's not the gold standard. It's the only way of isolating. You know if you want to get even more stupid, they call isolation, i.e., taking things and making them one, by adding more stuff.
Yeah.
Even that principle is on its head.
And so a couple more points, and one is homing in on a word you just used, which was stupid. And I want to impress upon the audience that I'm not anyway. I'm not going to speak for you. This isn't claiming some vast conspiracy that is happening within virology. I think that's really important to understand, because people will immediately leap to, well, why? Why would they do this? Sometimes we can take wrong turns. Sometimes science and all of humanity can take a wrong turn. And sometimes things like money can be introduced to a field where it shouldn't be. And it can start to direct that field and direct human behavior in a way that is antithetical to science and the scientific method. That can happen. It has happened in the past. It has happened all throughout the course of human history. And what we're saying is this just happens to be what's happening now.
Without a doubt, I completely agree that I'm accusing nobody of fraud. I don't think anybody's intentionally doing this, and actually working hand in hand for the past 8 months. It's been probably more like a year and a half, with some of the people involved in this project who have been in the industry for 30 years. Has been an eye-opening experience to me, because they've described and they've managed, bless them, to come out the other end and I think that they really suffer talking to me because I just see it through the eyes of a child. I'm just like, what's that? What does that do? How do you do that? And they just don't think about it. They don't, it's one of those things that you're you're a cog in a machine, and the machine tells you you want these results, and these people are paying for it, and that's it. Yeah, and he and a few of the others of the seasoned professionals that have been in this game for a career have explained all the same things, which is there's a big Overton window of methods. There's a big Overton window of protocol. And when things don't work the first time, it's just tweak a bit here, tweak a bit there, rerun it. They start with the end goal. The end goal is what they want it to look like, what they want it to be, what they know the results are, and they manipulate it up to that. And I don't think that they necessarily view it that way, that it's kind of, they are essentially, manipulating the results. They just know what they think is good science.
Mm-hmm.
That they have an image of, oh, this cell culture should look all dead to me because I'm knowingly putting in somebody that has tested PCR positive for COVID, for instance. So they know that the outcome should be over here. And if it's not, then they kind of just go, oh, just add a little bit more antibiotics, or just do this, just passage it one more time, and we'll get there, And so I agree with your sentiment that I don't look at these people, as anything more than just kind of, a cog in a machine that is doing their kind of job. And,
And that's another key phrase, is they're doing a job. Like any of us, we're taught. We enter into a career or a field of education. We are taught. We're not taught how to think about that thing. We're taught what to think about that thing, how to perform that task. We're given a paycheck. We are not incentivized to question the way that we were taught to do this thing. We just know that we show up to do our work, we collect our paycheck, and we try to continue that so that we can provide for ourselves and our families. I mean, that this is where my heart goes out to folks who are in this field who don't understand that they're no longer performing science. They're living for their paycheck. They're working for their paycheck. I mean, I wouldn't say my heart goes out to them necessarily. I want to see the human side of this, and I don't want to.
No, that's right.
And I don't want to paint this all as just this vast conspiracy that all virologists are in on. I think we're all human. We're all capable of making these mistakes, these wrong turns, proceeding in a life, making decisions, doing things just because that's what we've been taught to do. But before we jump into your presentation, Mike, I want to make sure that you get a chance here to weigh in before we launch.
No, it's fine. Any of the questions that have come up for me, you guys have pretty much covered. I mean, Jamie, I wanted to ask what really gave you the resolve and inspiration to even take this on in the first place? But you basically answered that. You're an honorable, upstanding gentleman who wanted to get to the bottom of something.
Oh, thank you. You're very kind. The scientific interest was there. I I had a sneaking suspicion. As I mentioned, my background was in geology. I I studied at university in the UK, and as I mentioned, they kind of did nothing to engage me right up until the third year, the very last year that I was there, and they gave us a project for data compiling of mass spectrometry there. And they had the most accurate paleoclimate data that came out of a frozen lake, Vostok, which is in Antarctica, which is their most accurate climate data. So what they do is they take mass spectrometry of these ice cores, and it contains oxygen isotopes. So there are two oxygen isotopes, 16 and 18, one's light, one's heavy. And to date, it's the most accurate way of mapping paleoclimates. So they claim that in hotter environments, the oxygen 16 evaporates off, and you get higher ratios of oxygen 18 when it's hotter, and vice versa. And as an undergrad, I compiled this data, and they also gave data out on CO2, because they did mass spec of the ice cores for that. We weren't meant to plot this data on the map, but I went ahead and did it anyway, just out of curiosity. And lo and behold, it showed about a 6 to 800 degree, 6 to 800 year lag of carbon dioxide to oxygen isotopes or paleo temperatures. And I pointed this out to a lecturer. I said, are you aware that, it can't possibly be that carbon dioxide is a driver for paleoclimates? And at the time we were, well, not at the time, the department of this university was sponsored by ExxonMobil. And I remember to this day his just blank face, and he just tapped the folder that had ExxonMobil written across the front. And so I was very aware that of basically, what is the UN, which is, the IPCC, the International Panel for Climate Change. At that point, that was in 2000, that I'd known from then that the climate change thing has categorically been a hoax. And so I was kind of aware when it came into 2020 that it was totally possible for such a hoax to be perpetrated on a wide scale. When it came to virology, it wasn't out of the question for me. So I guess I had a lead up to it. I guess I had an introduction, that I was starting from a base point, that I knew that it was perfectly possible for them to basically lie on such a grand scale.
Yeah, especially in such massively centralized systems, which I think at this point in time, we all, for lack of a better term, take for granted. Well, that's just the way the world is. But when we decentralize, which is exactly what you've done, we arrive at new conclusions. So before you jump in, I just wanted to ask one more thing, which is, and you basically have answered it, but it sounds like you didn't really run into any resistance from these different labs you were working with, because like you said, you hire them to run a particular procedure and they do it, kind of no questions asked. And it's, again, for lack of a better term, it's not that they're so much agnostic during the proceedings, but it sounds like you actually had some converts along the way as well. Is that correct? Is that fair to say?
That's correct. I mean, there's two kind of sides to this project. We're commissioning the work from CROs, and obviously they're there to do a job, and so we're not kind of converting them. Where you can't just approach them and say, do the science thing, mate. Here's the money. You have to obviously know some of the practice and the jargon, and there's
Right.
a to and a fro, but
Yeah.
they are research laboratories. That's what they do for money. And so as long as you're in the general vicinity, they will accept anything, any commissioned work. And that's the beauty of it. It's very easy. But there have been plenty of, and actually, I kind of, in a way, it may sound a bit masochistic, but I think that 2020 was a blessing. 2020 was very good for opening up people's minds and eyes, and mine too. I've learned something invaluable, which is contagion is a myth. And as you say, there have been numerous people, and I've got in contact and work closely with seasoned professionals that understand the same, that contagion is a myth, and that have gone along that same learning curve as me. And it's only growing as well, which is great.
Amazing. Well, let's dive into the presentation. For the folks who are just listening, we'll do our best to describe what we're seeing. We do recommend you go over and check this out on YouTube, because visual component is pretty important here.
Yeah, so here we conducted 12 experiments. So we took Stefan Lanka's work and repeated it 12 times, and we took in over 90 cell cultures. So we set ourselves the highest hurdle to achieve. We wanted to take this procedure and choose the hardest materials for us to control. Usually, and in Stefan Lanka's work, they use vero cells, monkey kidney cells. We chose to use human embryonic kidney cells for a couple of reasons. The HEK293T cell line, because they are the most widespread in clinical science, and they are also known as very robust. They're harder to break down than a vero cell. So we wanted to give ourselves the highest hurdle to control, the strictest parameters to try and breach the threshold so that we could steel man the results that we were getting. And also we used them because they're human as well. I mean, some of the things that they use different cell lines to isolate different supposed viruses, and they isolate supposed SARS-CoV-2 in a vero cell line because they effectively can't get it to break down HEK cell lines, human kidney cell lines. Well, even in vitro, and this obviously has no semblance on real life, as soon as you take a cell away from a body, you have no idea that it's acting in the same way. So this is, this whole experiment is complete pseudoscience, but they claim that it mimics a body. But, when they're isolating SARS-CoV-2, they use a monkey kidney. Well, even that process, it's got nothing to do with humans, does it? So here we're using a human cell line to show, the results of that. So here we have experiment 6. Here at the top, you can see this is 10% fetal bovine serum. So this is a percentage of fetal bovine serum which is considered nutritious. So it's the growing phase. And this operates as our negative control. Yes, we have controls in our control experiment. Don't you worry. This is a valid scientific experiment. We're not doing the same as virologists. Here we have the growing cell line. We grow it out to what's called confluence, where it has a happy and healthy medium, and it grows to, and here we can see on the right-hand side, we have readout, and it says live 95%, dead 5%. Let me just get, where's, I can't get, anyway, I can't, it says 95% and 5%, which is a readout of Countess, which is a machine that we've used to objectively verify cytopathic effects. Now, obviously, what virologists do is they see these things occurring and they push everything into their realm. So they just say, I determine what is classified as CPE or not. Whereas we wanted something that was objectively verifiable. So somebody that, or something in this case, which is a machine called Countess. So if you can see at the top, here's a link to the Thermo Fisher website, which describes more about what the machine is, but it's basically laser spectrometry, which counts the amount of live and dead cells in a sample. So it objectively verifies cytopathic effect for us.
And
so,
actually, Jamie, let me just interrupt real quick because I don't think you mentioned cytopathic effect earlier when we were talking about cell cultures. For the audience, just a reminder, when the virologists look at the cell dying and say, aha, that's because of the virus that was in the sample that we gave it, that dying of the cell is the cytopathic effect. You'll also hear it called CPE. Sorry to interrupt. Continue.
No, that's great. That's correct. Yeah, it's described as apoptosis, cell death, cincture. There's lots of kind of morphological changes, but as you'll see from the slides, it's pretty distinct. It's unmistakable. So here we see, 95% and 5%. So that shows that the cells have grown out to 95% of the dish. They're alive, they're well, but they're not overcrowded. If you overgrow them, which is one of the problems that some people had with Stefan Lanka's work, and we took Stefan Lanka's work and wanted to expand upon it and steel-man it as well. So we were running with strict guidelines to make sure that the cells weren't overgrown with the CRO that we used, and it came back with 5%. So it shows that they're not overgrown. So here is the one of the experiments below. You see we've removed the nutrients to 2% fetal bovine serum, which is following the protocol for, we used the adenovirus protocol. So HEK cells are typically used to isolate quote unquote adenoviruses and lentiviruses, just to random viruses, and we followed their protocols and they take them down to 2%. And here we see at day 4, if you can see, and I'll read it out, we have a count-test measurement of live 66 and dead 44%. So we have objectively verified 44%. That's nearly half of the cell line has died within day 4. And if you can see at the top, we have 1 times pen strep. Now, pen strep stands for penicillin and streptomycin, which are 2 antibiotics. They're the lowest grade antibiotics. They are less damaging, but still damaging, but they're less damaging than gentamicin and amphotericin, which we also did include in these control experiments. We included amphotericin to see the differences. But here you can see 1, 2, and 3 times. So we've passaged according to protocol, and we've put in 1, 2, and 3 times penicillin and streptomycin. And as you can see, reading from left to right, it says dead 32, dead 30, and dead 44. It slightly decreases, but it's variable across the culture. So you get the general idea that from the left to the right, 1 times to 3 times is 32 to 44% dead, which suggests that the removal of the fetal bovine serum, as well as the incremental increase in antibiotics, is causing the cytopathic effect. So we'll move on to the next experiment here. Again, this is at day 4. So we experienced cytopathic effect, apoptosis, cell death at days 2. It started to appear, and we took the photos at 20 times magnification at day 4. Here we have just tried A/B, tested it against 1% fetal bovine serum, which is the bottom set. 1% is not really used too often within many protocols. It is used in some protocols, but again, we wanted to get as much data as we could out of this control experiment, and so we just A/B tested it. You can see there's a little bit of change: 32 to 35, 30 to 36, 44 to 36. It's all kind of roughly the same amount of cytopathic effect. So that shows that it's basically, the biggest difference is just the initial starvation from 10 to 2%, and then from 2 to 1%, they're basically starving and they will die regardless of what's occurring. So here we had another control which acted as, I guess it's a negative control, but it could have been a positive control. We chose, and if I could kind of clarify on this, because some people say that effectively you can't control what is a pseudoscience because you've never identified the independent variable in the first place. You know, because they've never actually purified and isolated this thing in the first place. How can you actually conduct a science experiment? You can't control pseudoscience. Well, I kind of disagree on that front. And I've explained it in the Substack that I've put out that I'll give you guys the link to at the end. But if I could kind of go back to the analogy that I gave before of a control experiment where we have the ice creams, the sunburn, and the environment, those all objectively exist. But what if you took something that objectively doesn't exist? So you claim that a unicorn causes sunburn. So somebody's run out and they've said, actually, it's not ice cream sales, it's unicorns. Definitely. I saw one the other day. He was hanging around the pier and he was causing all of the sunburn to occur. So you can just rerun the experiment. You rerun the control experiment. So you take the beach, you take the controlled environment, you take this in the summer, in the sun, on the beach, you make sure that there are no unicorns in the vicinity, and lo and behold, you have sunburn. So you've proved that, again, unicorns don't cause sunburn. It's irrelevant that unicorns don't exist. It's irrelevant that essentially you are dealing with something that there is no independent variable. You have still produced a scientific experiment that has controlled its environment, and that has repeatable results, and that at least one of the variables is real. So I would argue that you can control practically anything. Obviously, you would get into the weeds if you had no variables that were real, where you had no observed effect. And to some degree, that's occurring here because it's not related to real life. This is all in vitro in the Petri dish. The observed effect is cell death, and you're attributing cell death to what is disease effectively. But
now, of course, one of the criticisms potentially could be that, well, a virus must have gotten in somehow, and that's what caused the cytopathic effect, or a bacteria would have gotten in here somehow, and that's what would have caused this cytopathic effect. And you're trying to protect against that, essentially.
Yeah, I mean, as I mentioned before, we are releasing, a very comprehensive methodology, way above and beyond any published and peer-reviewed paper, whereby we video and we document every single step of this. And we show chain of custody where we're sending samples off by scientific recorded posts in, sealed boxes on ice. So we're, doing everything according to the book, according to virological processes, and documenting it. So, if there's been claims that, I mean, as we'll see later on, we're identifying HIV, measles, and SARS-CoV-2 in the cultures that can't possibly contain any viruses. There's been claims that potentially these cell lines, have got them in already. Well, when you buy a cell line, and that's what we're doing, is buying a cell line from the ATCC, they guarantee that they're negative. So they PCR test them, they sterilize them, and they send them out to you because they're done in clinical testing around the world. This is the bank where you buy them from. And the notion that the ATCC was sending out cell lines that had HIV, measles, and SARS-CoV-2 already in them, not only is absurd, but it falsifies the entire, it's job done for me. If you want to admit that these cell lines all are contaminated with these things, you ain't ever going to find any, reputable data because they've already got them in already.
Right.
You ain't ever going to, isolate polio because it's got, how do you know you've got polio and it's not all HIV? You get what I mean? So it's already falsified. If you want to claim contamination, you've shot yourself in the foot really with this, right. Because we're using the standard protocol to get it. So, I think, and we welcome all of these, questions and firming up, because that's what we want. We want to convince the layman. We want to convince people that this is the truth, and we want people to understand that this is the truth. And that's the way to do it, because, you know, you can't just go up to a virological lab or Anthony Fauci or anything like that and go, what was your HEPA filters? You know, we're using proper HEPA filters in the thing, but we want to make it open. That's stupid that you come up to us and just go, I have a question, you know, that, you know, would convince me that what you're doing is just logical. It's not even like scientific. It's just as long as it makes sense in your head that it's logic that we're carrying out science, that's what matters to me.
Mm-hmm.
And so here we use sputum in this, and we use it because, you know, if we're adding, and people have suggested, and I think we will do it as well, is to add what they consider to be a viral titer. Now, obviously, we have, even in the last slide, falsified the isolation process, because we've proved that within 4 days, which is within the remit of calling something having a virus in it, according to the American Society of Microbiology, they claim that within 5 days, and there's a slide later on that I'll show you, but within 5 days, if you have any cytopathic effect whatsoever, you have a virus in your culture. So we have shown, and we have falsified this area of science. And so if we're to add a viral titer, quote unquote, viral titer, that comes from the cell culture. Okay, so it already has antibiotics in it. It already has reduced medium in it. So you would never know, even if we added this viral titer to it and the cytopathic effect was 100% in 4 days. I mean, it never is. It's about 40%. It's about this much. Sometimes a bit more, sometimes a bit less. It depends what cell line you're using. It depends on, you know, how long you leave it. But, you know, within these days, that's kind of what we're getting. So I wouldn't expect it to do it anyway. But even if it did, even if it went bang, it's 100%, we have still falsified this so that we know that if you were to add a viral titer, it would contain things that knowingly have already been falsified by causing cytopathic effects, cell death and apoptosis on their own, i.e. the antibiotics and reduced serum. So we chose to use sputum from a healthy, non-symptom displaying male into our cultures to see what effect the possibility of inclusion of you know, what is uncontrolled human sample into a thing would do. So you have all of the ability to introduce, you know, quote unquote contamination into a handful of these samples. And as you can see, I'll read it off again for the audio listener. At the top, so this is acting kind of as our control, our base at 2% against our sputum. We have dead 34%, live 66 in our top at 2% fetal bovine serum with no sputum. With sputum, we have 65% and 35%, so almost exactly the same. Right. So we're seeing basically no difference with addition of human sputum in it, which kind of rules out. I mean, it would be good to conduct this experiment with a disease, you know, symptom showing person, but you know, they claim that viruses are everywhere, right? They claim that there's trillions of them just floating around all the time. So you would expect if somebody was to gob in it, that it would have some, you know, quote unquote pathogens in. And that's what we've done here, you know, to kind of display that the inclusion of human sputum does nothing to the cytopathic effect.
Yeah.
They also claim asymptomatic spread, lest we forget.
Yeah, well, that's it. Sorry, it wasn't a symptomless person. It was an asymptomatic person. He felt absolutely fine, yep. But he could die tomorrow or in 100 years.
Yes, thank you for clarifying that, Jamie.
So moved on here, I described the American Society for Microbiology. And if I can just read out for the audio listener what we have here. So this is straight out of the manual for cell culture protocol. And it says the rate of CPE, cytopathic effect, appearance is also a characteristic that can be used to help identify viruses. In general, the rule of thumb is that a virus is considered slow if cytopathic effect appears after 4 to 5 days in cultures inoculated at low MOI, and rapid if cytopathic effect appears after 1 to 2 days in cultures inoculated at low MOI. So what they're effectively saying is that at any point, if cytopathic effect appears, and that's not, the huge amount that we've experienced, just starts to appear. So in this experiment, it started to appear at 48 hours post removal of FBS, and it rose to nearly half dead at day 4. So if you're to look at this, we have a virus in our culture, according to the American Society of Microbiology, because within four to 5 days, we have a cell line that's half dead. So according to them, we should be going to them going, I don't know what virus it is in our culture, but we have a virus. So here's the bottom, just to firm up about the cell lines, the HEK293 cell lines, just to show you that generally regarded as a robust and low maintenance cell line is what it says here from the bank, from the cell bank. And here's another cell line, that they use. The HUH7 cell line is a permanent cell line established from male hepatoma tissue. So that's a lung cancer tumor, which was surgically removed from a 57-year-old Japanese male in 1982. So every single cell line that you get was from this one bloke who died from liver cancer, and they freeze them and just grow them. But it shows just how absurd it is that they claim that when they remove tissue from a dead old Japanese bloke, they keep it in a freezer for 40 years, and then they unfreeze it, and then they put it in a petri dish, and then they starve it, and it dies, and they go, that's because it's a virus. It's like putting that 57-year-old bloke with his hands tied behind him in a boxing ring with Mike Tyson and going, right, it's a fair fight.
Science.
That's how absurd it is. And when you actually start to kind of pick away at it becomes farcical.
Mm-hmm.
So we finished that part of the cell culture, and we sent those cell cultures that we had off to an independent microscopy lab, and we gave them a brief of find the extracellular vesicle. We were funds limited at the time because I was just looking to keep this kind of private amongst a small group of people. We funded it, and we didn't have enough money to choose their premium package, which enabled us to basically have an open scope conference call with them. So what they have to do is they fix it. So they kill the cell line. They fix it in a formaldehyde type thing, a really noxious substance. And then so basically, that means that you can't see any movement. Hint, hint. It's impossible to see anything going into or out of a cell. Hint, hint. Mm-hmm. We could have, with their premium package, looked through our cell cultures and pointed them at what we wanted photographed, but we didn't have enough money to do that, and it's very expensive. So we got them we gave them a brief to find extracellular vesicles and report back to us the findings. And they took 9 photos. And this is the first 9 photos. We have conducted no other TEMs of these cultures ever. The first try with 9 photos, and 3 of those you'll see on the screen were of extracellular vesicles. So the independent CRO, the research lab, took the microscopy pictures and sent them back and said that in the middle, and I'll describe it for you, is completely empty. It's just a lipid layer surrounded bubble. Yeah. And I can tell you the graph for the size is just out of shot, but it's 2000 nanometers, which is a lot bigger than what they claim a virus is. So it's quite clearly not a virus.
Mm-hmm.
It's quite clearly not what they call a virus. It's much bigger, and there's been some contention it's incorrect, but extracellular vesicles can be, especially in apoptosis state, they can be up to 5,000 nanometers in size. So that's well within the remit. They've correctly identified that as an extracellular vesicle. And that's an important point, actually, is that they have identified something which we will go on to see something cannot be.
Okay.
So here at the top, we move on to the next slide, and I have put next to these are images from the CDC. They're images of what they consider to be Omicron subvariant BA2. Very specific, I know. And in purple, we have a round blob with these inclusions in them, which they claim are the proteins, and it has a kind of halo around it of lighter gray. And then around them, it has clustered vaguely similar looking things, which they have identified as Omicron subvariant BA2. Some of them with more or less of these inclusions, but they're all roughly round. They're all roughly have, between, I would describe, between 4 to 10 inclusions in them, and they all have that rough kind of halo. So that's what the CDC are claiming is an Omicron subvariant BA2. This, in the picture below, is our cell culture, which cannot possibly contain a virus. Contains what looks identical to their image above, which is rounded. It has these kind of protein inclusions. It has the halo surrounding it. Now, some people have said that, the halo in the top area is light gray, whereas the area is dark gray below. Well, that's just a contrast issue. They send out about 5 or 6 different contrasts, and I chose the more bright contrasts so that you could see it. They're grayscale, so you don't know what color it is, but it certainly has, a halo around it. It has those kind of same inclusions in there. And more to the point, the size is very indicative. So here we have a readout of diameter varies, and this is straight from the National Institutes of Health. Diameter varies from about 60 to 140 nanometers in size for SARS-CoV-2. That's how large it is. So here at the bottom, you can see the zoomed out picture of the transmission electron microscopy that we got. You can see a couple of the other similar looking rounded particles that are here. And here in the above picture, we can see, and I've cropped and I've edited it so that the bar scale that they were given to, let me just zoom down. You can see it just at the bottom right-hand picture here. I've essentially just cropped it and moved it up and then zoomed in so that you can get the exact size of this particle, which is about, 1/5. You could fit about 5 of these into that bar scale. So it's about 100 to 120 nanometers in size, which is exactly the size of, what they claim is SARS-CoV-2. Now here in the top picture, we have what the CDC describe as being a measles virus. So you'll see that it differs from what they claim is SARS-CoV-2. It's very distinctly oval in shape. It has these dotted inclusions. They're not so, central, circular. They're not so arranged in a circular pattern. They're kind of more sporadic and spaced out. And they even go into naming these things. They call them phosphoproteins and fusion proteins, and then they claim that they say it's in some kind of matrix, and then they say that there's a viral membrane around the outside. Well, this is our transmission electron microscopy, which again cannot contain a virus. You'll note that it has an oval shape, a similar distinctive shape. It has sporadic, they're not circular, it's not in any sort of pattern. They are sporadic inclusions in them. And you'll note that it has this matrix inside of it, and then it's surrounded by some kind of membrane to keep it together.
Jamie, JB, sorry, just to go back to the measles one, that top image above yours is an image from TEM, from electron
That's
microscopy?
an image from TEM, from the CDC. That's correct. Yeah,
it is. Okay. I would say there's incredible similarities between what the CDC shows as measles and what you found in your sample.
Yeah, and even more so the size. I mean, the size is massively important in terms of, identifying these things, because, when you're saying, and here I'll read it off. Again, this is from the CDC, measles virus. It is 120 to 250 nanometers in diameter. And so again, I've done exactly the same thing, where I've taken the whole scale picture, and I've cut out the size chart, and I've put it next to it, and it's half the size. It's 250 nanometers, so it's a large measles virus. So you have something which is oval. It has sporadic protein inclusion. It has the matrix. It is encapsulated, and it's exactly the same size.
Okay.
From these 6 pictures, at the top here, we have HIV, what the CDC considered to be HIV. So you'll note that they're round. They contain this kind of nucleus. It looks like a kind of fried egg is what I would describe it as, with a dark spot in the middle and then lighter around the outside. And with some kind of membrane around it. And here, this is from our culture in the bottom, which is round. There's a few in the vicinity. It has a nucleus, that kind of fried egg pattern, and it has a kind of membrane around it. And again, we'll go back to the size. So the mature, reading off of the National Institutes of Health, the mature HIV particle is round, measures approximately 100 nanometers in diameter, with an outer lipid membrane as its envelope. So here we have it next to 500 nanometers. And again, you might be able to fit that in 5 times. So it's 100 nanometers in size. It's exactly the same size. It has exactly the same nucleated inclusions. And it has the outer shell. So by looks alone, and this is the key point, by looks alone, we cannot say that there's a virus in there. Now, lots of people have kind of come to me and had initial rebuttals and said, but you haven't, genetically identified it yet. You haven't done all of that. No, we haven't. We're at that stage now. That's what we're doing at the moment. Mm-hmm. But what I'm doing is I'm moving through every single stage that the virologists claim as proof of existence of viruses, and I am falsifying it. Okay, so we falsified the cell culture. We said that they can't say whether there is anything in their culture because the effect is seen without there being a possibility of a virus in. I've moved on to TEM, where, and this is a big one for me, because they point at these things and they say, that's a virus. This is what they're pointing at. They're pointing at cell debris. They're pointing at when the cells die, they have these fat membranes, lipid membranes around them, and they just bud off like a lava lamp. It's just they're dying, and that hydroscopic budding off of some of them we saw don't have anything in them, any of this cell debris. Just look to the left, to the left-hand side at the bottom here. This is just the cell. It just contains loads of crap, like just cell debris. Yeah. All these things that they're pointing at are just bits of the cell, right? And virologists are pointing at those bits of cell and going, that's a virus. And all we're doing here is falsifying purely in microscopy that is not a virus. That's not a virus that you're looking at. Okay, because it's a huge thing on kind of the battlefield of ideas, online, on social media. It's one of the first things when it comes up, oh, I don't believe that viruses exist. Well, of course they do. You can see them, duh. You can see them. You put them under a microscope and you can point at them. No, you can't. No, you can't. Because as we've adequately shown here, within 6 photos, the first 6 that we had absolutely no control over, if we got, an hour to point at these things, we could find thousands of them. I have absolutely no doubt that we could tick off every single, quote unquote virus known to man, in these cell cultures. So, and that's in one cell line, who knows within all of the other cell lines.
Yeah. If I were the one doing this work, I would look at it and go, oh, this poor person, he's got COVID, he's got HIV, he's got measles.
Yeah, he's on death's door.
How is he even living?
And this is the scary thing is they can prescribe all these kind of drugs, remdesivir and AZT and measles vaccines and stuff for these people.
The use of electron microscopy has become one of the instrumental tools in selling this story, because seeing is believing, and then from the electron microscopy you get the like artist's rendition and the CGI and the Photoshop image of the virus, the sphere with the spikes out of it that looks threatening. It looks like something that's going to invade you, and this is all just part of the story building that
exactly
leads to,
exactly fear. For me it was huge, and I was delighted. As I said before, we knew going into the cell culture experiment what to expect. We had absolutely no doubt that when you starve a cell line, you get cytopathic effects. Whereas when we were sending this off, it was a bit like, oh God, are we sure about this? We spent $2,000, to send it off and get it back. If we just, spunk this money up the wall and we're going to get back a whole load of stuff and just be like, oh crap. But it was just stunning to me. It was stunning to me that open these 6 images and I was just like, hold on a second, that looks exact, wait, and that, it was just, it was just real affirmation for me, for actually going, for knowing truthfully that they're talking bollocks.
Yep.
And so, here's some of the feedback from when we released this information, as a public release on AlexX show a few weeks ago. There's numerous people online that I've kind of been interviewing and kept in contact that have provided a lot of, the rebuttal to Stefan Lanka's work that I, used to kind of give me their objections, from the other side of the table, as it were, because these people all believe that, viruses exist. They claim to be credentialed virologists and microbiologists, all of the people in these, Twitter threads. And this is what they kind of have to say. So K-State Turku is a microbiologist in the US. TEM is not an identity test unless it's coupled with immunostaining. Spend your money on mass spec and genomic sequencing instead. So there he's admitting that basically transmission electron microscopy can't be used to claim that viruses exist. And it says from Virology Truth, the lab states themselves that those particles could not possibly be extracellular vesicles. If they weren't extracellular vesicles, what were they? He says, I need to see the protein composition of those particles. He's not showing the data, whatever it, he's not showing the data because he knows it will make him look foolish. No, that's a lie. I haven't done any proteomics yet. We're going to do them, and we will release them when we find them. And what else could those particles, and here's the crucial part, what else could those particles be? And K-State Turk replies, I don't know. I don't know. That's why you need an identity test. And that's a very powerful thing. Just, I don't know what else they could be. There is no other explanation for what they could be, which is why they have to move it out of the realm and do PCR, do genomics sequencing, do all of the other extraneous stuff, which takes it completely away from the scene, takes it from the physical world, and takes it into an in silico computer model, because that's where they can function. Here we have Thomas Moore, who is a credentialed microbiologist. It's absolutely logical. They observe CPE. So what? In virus-induced CPE, one can detect the proteins in the genome of viruses. They didn't detect that. That means the experiment was a huge waste of money. We haven't tried yet, so we will get to that. Brendan replies, what virus caused their CPE? I.e., suggesting there was no inclusion of a possibility of a virus. So what caused the CPE? And Thomas Moore replies, CPE is caused by many things, starvation, for example. So he's just clarified there that, the protocol itself is causing the CPE. So even by their own standards, they know these things, contain, materials that cause the effect that they're looking for anyway. There's plenty of peer-reviewed studies out there that show this too.
Yeah, and you see what they're doing. They're just trying to distance themselves from the cell culture isolation, and then from electron microscopy, and saying, well, you still haven't done this and this. They're just wanting to distance themselves from the fallacies in those approaches.
Here's Sentstrand, Thomas Baldwin, who's a microbiologist as well, and deals with plants, so maybe not too highbrow, but I'll take his opinion anyway. He says, will you take that bet if Jamie's mistreated cells, and he has a running gag that we've mistreated these cells, produced a genome that resembles a novel virus, virology is falsified. And here's the key thing, is that we've got it to a stage where any intellectually honest scientist will agree that none of these processes that we have falsified can constitute as evidence of the existence of a virus. We cannot prove a negative. It's impossible to prove that unicorns don't exist, because there is always a possibility, even though it's a very minute possibility, that they do exist. So what we have to do is falsify the evidence that they do. And that's what we've done. And that's what any intellectually honest scientist would agree, that there is no evidence that viruses exist within the cell culture isolation. There is no evidence that viruses exist within the transmission electron microscopy. We have moved the conversation to the point where all eggs are in the baskets of genetics.
And that's where we're at
now.
So
I'm happy with where this project has arrived to, and that's why we have chosen to bring this project. We knew that the end goal is genetics. We always knew these two areas of science are dependent. I mean, virology is completely dependent on genetics.
Okay, so that brings us to the present.
That brings us to the present, where we are with this project. And the further studies that we want to do. So PCR, full transcriptomic sequencing, antibody tests, and proteomics. Now, antibody tests have already been falsified. There's a peer-reviewed paper where they try to debunk there were kids in the UK actually putting Coca-Cola on their lateral flow tests to get off school. It was amazing. Fantastic crowdsourced project. All these kids, yeah, just figured out how to junk these science experiments. All, you know, the big men in their white coats and their labs, and a bunch of teenagers found out how to, conduct basically what is a control experiment. They found out that if you put Coca-Cola on them, they gave a positive every time. And so the establishment had to scramble and do a control experiment to show that it was, it's acidity. They claim that it's acidity that's causing these things, off-label usage is what they called them. The problem is that they shot themselves in the foot, because they did a control experiment where they put still bottled water and they put vodka on. And vodka is actually, surprisingly, very pH neutral. It's almost exactly the same pH as the human body. And it's also sterile. It's sterile because of the alcohol content. And still bottled water is obviously still bottled water. Your body is made of 60% of water, 60% plus of water. And both of those came back positive, putting them directly onto a lateral flow test. So it's been falsified already. It'll take 2 seconds and we will buy a couple of these, pregnancy looking sticks and fetal bovine serum on them and get positive results. I have absolutely no doubt. So that area is, very easy.
Can you describe then what the further studies, can you just go into briefly here what exactly you mean by PCR, the studies that you're going to do, and then your approach, the methodology you'll take, and then the same thing with the sequencing?
Yeah. PCR is a bit more difficult, actually. You would have thought that the full transcriptomics was because, they're mapping the entire genome, whereas PCR is just taking fragments of it and seeing if it matches up. I don't know if you want me to kind of go into it, the basic principles. I mean, the fundamentals of this is that they take it away from the real world. What they're doing within genomics is they take a sample and they add a dye. They add a fluorescent dye knowingly into the sample, as if you're pipetting it into a sample. They then put a primer into the sample, which supposedly only allows this dye to fluoresce when a nucleotide sequence matches up with its respective nucleotide sequence. So obviously you have ATCG and they all map to their respective nucleotides. I don't know if I need to go into kind of genetics, but a primer is one side of that nucleotide. And so it claims that when they add it and it matches up with another, it has a thing called a quencher, which allows that to fluoresce, or the quencher comes off, detaches off. So that's stopping it. And it allows this dye that they've put in to fluoresce. And all a PCR machine is, all a genomic sequencing machine is a camera. It heats it up. It's got a heating element. It heats it up to a certain degree, which activates an enzyme in it. And then the camera shines down and it picks up how much dye is in these things. So if I was to simplify it even more, they are knowingly putting a fluorescent dye in, shining a camera, and when they pick up that fluorescent dye, they are claiming that there's a huge array of unseen, very complex things happening with no proof. You can't see it. You can't see a nucleotide. You can't see the strings. You can't see the strands. You can't see them together. It's completely unseen. So they're adding a dye. They're seeing a dye and claiming there's all of these hugely complex processes in between, which is just stupid on the surface of it.
Mm-hmm.
And so when it comes to the control experiments, actually it's a whole lot more difficult with the PCR because there's a lot of moving parts with it. If I would say that the oligonucleotides that they use for the primers, the positive control, which is the thing that they make these things in chemistry. It's all chemistry, and my chemistry is a bit rusty, so I kind of need to brush up on it through these periods. But there's over 200 chemicals. There's over 50 methodological processes that go into making this thing, which supposedly mimics a nucleotide sequence. 200 different chemicals. None of them are controlled. None of them are, why is that in there? What does that do? They just come out and they make this thing. And then, so the PCR process is quite difficult because the output of it is just a positive or a negative. It's done on cycles, how at what cycle threshold this thing amplifies. So when you can set, there's a lot of moving parts within the machine as well, which enable you to set a threshold. So we're doing the genomic sequencing first, which is a lot easier from our perspective because there's so much human input. Because what they do is they take the primers, these very short reads, and they amplify all these reads. And there's plenty of information about how alignment works. But what they do is they amplify it with PCR, and then they take all of these short reads, like short letters in a book. Yeah. And then they have the book at the end that they want it to look like, and they just assemble it into as close to the book as possible and call that the genome. Well, all of this is done inside a computer program, all of it done with parameters which are all changeable and modifiable by a human being. So basically the genomics part is just finding, and controlling that part of alignment, and also finding out. And I mean, I can go into, some of the things that give you kind of an indication as to just the inaccuracies of what we're dealing with here. And it's pretty stunning. I mentioned it on Alex Zek's show, but I can kind of go into a bit more detail on here. This is that you might be surprised to find out that there are no blinded accuracy studies of human genetics, like forensics. Let me say that again. There are no blinded accuracy studies of human genetics. Barry Scheck, who was a lawyer in the Dream Team for, his name escapes me now, the glove bloke, just recently died.
OJ.
OJ Simpson, thank you. He was a lawyer in the OJ Simpson trial, and he was so disgusted with the inaccuracies that he was seeing with forensics data that after that trial, he set up his own organization called the Innocence Project, which was looking to back people who he believed were incorrectly accused or falsely found guilty of crimes through forensics data, through shoddy forensics data. And he went to the three-letter agencies, I think it's called the National Institute of Technology in the US, which is the accredited auditor of forensics data. And he asked them, have you got any blinded accuracy studies of genetic forensics, i.e. like, where you take a pool of 100 people and I know, that there's a known outcome. So there's a known person and we send in these 100 samples and you find the perpetrator that we know it is. And they said, no, that data doesn't exist. So he made them do it. He made them do a 3-person mix. So he took the saliva of 3 people, put them in a Petri dish and sent them off to 108 different labs and said, we know the perpetrator. It's either perpetrator 1, 2, 3, or they're not in the vial. Can you find the perpetrator of this crime? And out of 108 different labs, 6 got the right answer. So they tell you that genetics is 99.9% accurate.
Mm.
Oh, yeah, we know exactly what we're doing. Oh, of course, we can identify anything, fart in a breeze from a mile off. We'll sniff it out and tell you who's done it. No, no, no. It's less than 10%. That's less than if they had just fucking guessed.
Yeah, that's very frightening.
Yeah. And he went on and did it another time. He actually went and represented somebody at trial, whom he thought that the judge was busy mates with the geneticist, because obviously they all have their kind of go-to scientists, to kind of bang the gavel and go, you're guilty. And so they commission these genetics labs to do it. And Barry Scheck believed that they had just kind of made up in their own mind that this guy was guilty and fudged the genetics parts to it. So he took the guy's samples and sent them off to 18 different labs. And one came back with corroborating results of the initial decision. Again, 1 out of 18. It's less than a good guess. It's less than 10%.
Mm-hmm.
I mean, that's not even science. You're not even talking about science there. In statistics realms, that's the opposite of there being any sort of correlation. That is a negative, correlation. You are not. It's not even that it's accurate. It's that you're wrong. Do you get what I mean?
I do. Yeah.
And so what we're dealing with here, on the face of it, I'm quite confident. I'm quite confident that, and I don't really want to go into the realms of forensics, for obvious reasons, that some of the ramifications that it opens up is not really anything that I want to be involved in, to be honest.
That's a giant can of worms you'd be opening, and many cell doors potentially.
Yeah. But
so the idea would be that you would take your control experiment, for example, and you would find the genetic sequence of a virus in it when there's couldn't have been any
we're kind of
virus
fleshing it out at the moment. I don't want to give away too much because there's a lot of us, kind of involved in this project, including yourselves, including your good selves. There's 100 people kind of involved in this project, and we're all chipping in, and we're all doing our great work. And thank you for, I haven't said it yet, but thank you for interviewing me and getting me on. It's great to have the message out.
Well, great to have you on. So next steps here. Yeah, next steps. This is where the listener can get involved, correct?
Yeah. That's it. So we're planning to put together a lab with a thermocycler, which is the machine behind a PCR, core lab equipment to order, test PCR samples and software, as we're unable to send off the exact controls we want. Some of this is the reason really why I was explaining that PCR is more difficult, because to control it, if you're sending them off to a CRO, you have to send off very unorthodox things to be able to control it. Like, we just want to send off just positive control, or just primers with no sample in, or just sample with no primers or no control. Or do you get what I mean? These are very unorthodox. So approaching CROs, they will look at you like you're a madman. They just won't carry out those types of controls that we're asking for. So it makes it very difficult controlling this. We're basically going to have to buy a machine and do it ourselves, which is what I want to do. And I want to form a lab with the accredited microbiologists and virologists that I'm working with hand in hand, for a couple of reasons. That it enables us to do a lot of work. But secondly, and more importantly, is that when we get to the end of this and we falsify every single area, all they will do is they will change a part ever so slightly in the methodology and rerun it again, which is exactly what they do with vaccines. It's exactly what they do with drugs. As soon as they get found out and they get taken to court for drugs damages, vaccine damages, where harm is proved, they change one tiny ingredient and substitute it with another chemical that they made that just isn't the same chemical, but that is probably just equally as bad. And then they send it out again. And so we don't want to be put in a situation where if, 2020 happens again and they start, coming over on all strong again, with their, mythical mandates and, things that they're trying to coerce you into doing. We want to be able to be live so that we can take their methodology and refute it as quickly as possible. So that's kind of the reasoning for setting this up live, and what we want to do. In the short term, we have everything ready to go for full transcriptomic sequencing. We've actually started to send those off now. We've engaged numerous CROs to do that. And we just need some funds to kind of keep going with that. We would like to further the first two observations experiments by buying more cell lines. We want to conduct it with Vero cell lines. We want to conduct it with lung epithelial cell lines. There's a handful of the main ones that we want to kind of A/B test, and vials of what they consider to be virus to just see if there's any difference. Again, just the more data, the merrier. We would like to offer the chance for people donating to propose more control experiments. There are actually hundreds, maybe thousands of different variables that we could have within these things and other places to look at. And if you wish to donate funds for materials, we hope to carry out your requests in order to put another nail in the coffin of this pseudoscience called virology. And that's, we want to work hand in hand with people, to be a part of this project. It's not pay to play, obviously. It's just if you feel like donating and want to see something specific, that would be awesome, and we'd be totally up for doing that. And what we're sharing with this project, we actually have a product. Okay, we have a product that is the full methodology, the video of all of the methodological processes that have gone involved in it. We're also making video material of how to use this scientific document. So we're sharing all the methodology, materials, chain of custody, sourcing of materials will be written up in a scientific document that we're making open source. And that's the key to this. I've verified with lawyers online, and well, lawyers involved in the project, that you can make a scientific experiment open source. And when you make a scientific experiment open source, you can give it to people and they can use it in their own capacity. So you can assimilate it into your own material if you want to try and get it published, if you want to try and submit it to a journal, if you want to submit it to anywhere official, you can have it as your own. You can present it as your own data and say, this is my data and this is what I'm doing with it, expand upon it, or just use it in its own right. We'll be doing video explanations on how to use this document. It can be used, for example, as the likes of Lanka and Haberland and others winning based on the no virus claim in court to fight against employers pushing vax and mask mandates at international borders as in private spaces to prevent 2020 ever happening again. So you are presented, I'm in France and I had to get back to visit my parents in the UK, so they made me do those stupid, up the nose jobbies for PCR tests. And, I didn't really have any other way around, getting back. I had to go and see my parents, my children had to go and see their grandparents. And I want to empower people to have that scientific knowledge and that legal knowledge, because a controller operates as a legal vehicle. It is the legal vehicle for, Pfizer have been found guilty of scientific fraud and, falsely advertising drugs and the harms that drugs have been causing to the tune of over £10 billion over the years. And when it's not marketing, it is defrauding essentially what is the control process. The double-blinded placebo-controlled trial is what is considered the way to test a drug, right? And every single time when it comes down to scientific fraud, it is either fabrication or manipulation or wholesale leaving out of this control data. So as you can see, in the eyes of the law, a control is scientifically binding. And that's quite an important point, that when you're looking to back yourself, legally trying to back yourself against employers. So if you've been unfairly dismissed, if you felt coerced into taking a vaccine and been injured, I want to enable people to actually, I watched a few of the law, a few of the court decisions unfold. There was one in the UK, a guy called Simon Dolan, who actually lives not too far away from me in southwest France. But he owns a jet company, really, really wealthy bloke. And bless him, he tried to take the UK government to court on lockdowns. And he lost because of asymptomatic transmission. At the time, I was just kind of learning all this stuff and, wasn't really kind of up to speed. But, it's that kind of thing where, the virus is used as a get out clause. Oh, and somewhat I see it as a problem with a lot of the anti-vax movement as well, online philanthropists, that are anti-vaxxers, that, it's great that they're raising awareness for anti-vax, but if they still believe in the virus, it's the biggest get out clause, because all that people do is, they show this data, they show where, this huge peak exactly after vaccination. And all anybody does get COVID. Yeah, it's easy. It's easy. It's just the get out clause. And so this is what I want to empower people to be able to front with and be able to rebut with in these kind of areas.
It's pretty daunting for, I would say, a person who's just being introduced to the no virus position to wrap their heads around this, because the implications on the way that the world works are bigger than any other. This fundamentally changes the way that you move about the world, and the world moves around you.
That's right. I think it's, this project is not only that as well. It's not only virology. As I said, I came from kind of a geosciences background and, climate change. And, I think this is an important point, that if you understand what a control is, the power of control and the power of crowdsourcing science and decentralizing science into projects for finding out the truth, there's no reason why people can't do this for, climate change. There's no reason why people can't do this for all of these, pseudosciences that are being presented as cover for tyrannical government, policies. So that's what I want to encourage, is people just to formulate these groups and find out for themselves. And when it comes to mass, it would be mass spectrometry when it comes to geology or any of these other processes. So the implications are kind of much wider. So with this project, we've been working closely with Alex Eck and his company, The Way Forward, throughout the project behind the scenes, but launching it, he's very kindly hosted the project and is doing all of our admin, which is really lovely. It's lovely, bless him. And he's just done his website up as well. But if you do have any donations towards the project, that would be very much appreciated. Everything going to The Way Forward, it's www.thewayforward, and forward is FWRD.com, and then /donate. Every single penny that goes through that site is to the project. I have had a few people come to me and contact me personally saying, I'm just not sure because it doesn't say that it's explicitly to the project. We are making a landing page which says that. But I trust Alec. He has accountants dealing with all of the money, and we can give shout outs and show that people, their money has been received, and also where it's being put towards as well. Materials list exactly the projects. If you want it to go towards a specific thing, that's more than welcome. And, just come and speak to me privately.
Beautiful. And we'll put the link in the show notes of this episode.
Thank you. That's really great. Thank you.
Jamie, do you want to stop presenting just so we can get you back on screen? There we are.
Hey,
the line.
Sorry, behind the I got PTSD from doing the PowerPoint presentation. It's kind of taken me back to university times. It's just like, man, I forgot how boring this is.
Well, you nailed it. You nailed it. Well, I think maybe you've put your finger on one of the issues with science. Is I think maybe too many people find science boring, and perhaps we get overwhelmed by the phrases and the language that's used, and our eyes can glaze over. And this is what we've done now over the past 100+ years. We've just said, you know what, leave it to the experts. I'm going to trust the experts. And what is required of more of us, the lay people, is an investment in time and interest and attention into science, or else will continue to be pulled over our eyes. And those who we've entrusted to make these decisions based on science will use it for wrongdoing and for the wrong purposes, and to usurp power and control, and aggregate power and control, and keep us where we've been the last 4 years, which is like in the dark and under lockdown and all the rest.
I agree completely. It's just it's about taking responsibility for yourself, and finding out about your environment, and how that relates to your own health. That's the thing. I mean, government policy just in its own realm is farcical when you try and apply it to numerous people. Everybody's different, and when you start to look at disease and everything like that, and get really into the nitty gritty, I believe that I'm always very skeptical about any area of science where people say, you've got to eat this, or you shouldn't eat this, or do you know what I mean? There's lots of nutritionists out there who say, you've just got to cut, meat out, you've just got to cut vegetables out of your diet, or you just got to, only eat meat. And I just think it may work for some people, but I think any of that carte blanche, thing is a dangerous road to kind of go down. And I think that people need to conduct their own science with their own, bodies, and find out what works for them, and move like that.
Yeah. N of one, as they say.
That's right.
Yeah. Mike, jump in here before we wrap up.
I just want to say thank you for your due diligence, Jamie. It's almost like we've all heard, I think, especially our generation, about this supposed promise of the internet. And I think the work that you're doing is maybe the first time I've seen it manifested to the purpose that promise we all hoped would hold. Obviously, the governments and the big guys and the centralized powers had a head start on us, but you're really taking the bull by the horns and saying, what can we do with this information highway, with the ability to correspond and communicate, and not just do interviews, but contact labs and get the 6 photographs, and how much you can do with just the data war, for lack of a better term. So thank you for taking the bull by the horns and doing this work and sharing it with us all, because to me, like I said, it's the manifestation of the promise of the internet, and it's happening in real time today. So thank you.
No, thank you, Mike. That's it's really well put. And I describe it as kind of like a Bitcoin or something like that. There's been an answer of decentralization in a lot of other places. Bitcoin is the decentralization away from central banking, to make sure that they're not, deflating your money with quantitative easing, and people, and it's done remarkably well for the people investing in it, and it will continue to grow and decentralize, and it's fantastic. It, provides all of those areas to move away from the government and move away from all of these things happening. And I think that, you know, all of these new things had happened, but not to science. And that was what was kind of making me think, is like, why is it being conducted this way? And it's just purely a philosophical thing. It's purely a, you know, a small mental change, because all of the infrastructure is set up. All of the infrastructure, it's just bottom up rather than top down. And you know, obviously the complexity, and I hope that it grows as well, that I hope that, you know, it takes on its own form as well, in the way that, you know, things happen, you know, because, I mean, for instance, it's like, you know, all of the virological equipment and everything just gets junked once it gets proved. But I think that hopefully it can get used for good as well, you know, to actually better people's health, you know, whereby, because I think a lot of people want to, you know, they've been disillusioned with science because there's so much fraud, because there's so much bollocks talked. I think people have just wanted to throw the baby out with the bathwater. They've just gone, okay, well, it's done. It's like religion. Do you know what I mean? It's kind of, you know, bang, you know, onto the cart, and we'll kind of move on and we'll just look for something new. But I don't think that we should wholesale chuck it out just yet. I think that there are lots of processes and lots of accuracies and lots of methodologies that in principle can lead us to the truth. And I think that there's going to be a lot of stripping away of the fact, but essentially there is still a core principle around science that can be of benefit. And I want to keep that and grow that.
I think that's beautifully put. And I like to remind myself, especially during, like, you know, when Occupy Wall Street was sort of top of mind for everybody, there was this 1% versus the 99%. And there's so many people who resonate with that message. It's a powerful message, but we still have this like baggage of deferring to authority that we haven't been able to kick off our back through the lens of taking ownership of how much power we have. And that's really what you're doing by leading by example. Is the bottom archy, you know, the lower archy, whatever you want to say, whatever the archy is, you're making it not just a slogan and a bumper sticker, but like action. It's actionable. It's actionable decentralization and knowledge. And as Ben has pointed out over the episodes, when asked, or maybe early on, Ben, I wonder if you would still say this, and you probably would, that when asked what terrain theory is, you would say freedom from fear. And that's basically where you started this conversation, is once you can release the fear around the boogeyman virus that's never been proven to exist, then you have no choice but to take inventory of the things that can actually make you sick.
Correct.
And then it becomes very obvious. It becomes like kindergarten level obvious what does and doesn't make you sick. And so that's what you're helping through all your you know, hours and hours of work. We're getting to something very simple. And as we've often differentiated, you know, it doesn't mean it's not complex. It can still be complex getting there, but at the end of the day, it's very simple. So really, thank you so much for the Herculean effort you've put towards arriving at this simplicity that we know is inherent to what we're all driving at.
Thank you.
Jamie, I know you don't want this project to be about you, and we're grateful for that, but we do need to bring this back to you as we wrap up this episode and ask you the question that we ask all of our guests, and that is, what are your non-negotiables or the daily habits that you do to tend to your terrain?
You know, I'll continue the theme of just, you know, falsifying, like I don't kind of prescribe to find. I haven't applied myself to finding out exactly what causes disease. I think it's something that should be kind of, I want to get to the stage where we have accepted that contagion is a myth, as a, you know, as a populace, and then move forward to kind of find out together. And so I kind of treat my body in the same way, that I just don't take any pharmaceuticals ever. My whole family don't take any pharmaceuticals, no supplements, no nothing. And I try to eat as healthy, grass-fed, organic food as possible. And I'm building my house here with my hands. Everything is done myself. So I keep fit in that realm. I try. I believe in no extremes. I believe that you can exercise too much, for instance. I just think that exercise, if you do it, should just be a modicum. I think that, uh, I think that extreme diets are bad. I just think have a little bit of everything and see what works. And so that's how I treat my body on a daily basis.
Beautiful. And then last question, Jamie, where can the listener learn more about you and your work?
Yeah, I've just recently started a Substack, Jamie Andrews. Does it have an @ on it? I'm not entirely sure, but I'll give you the links at the end there, and I'll be releasing all of the methodology and the videos coming up very shortly. The video methodological processes on there, and I'll be giving kind of updates to the control studies. And I'm most active on Twitter at Jamie _A again, because I got banned quite a few times.
And we'll put the links to all of those again in the show notes, as well as the donate page on Alex, the way forward.
Fantastic. Thank you, guys.
Yeah, Jamie Andrews, thank you so much for joining us on the Terrain Theory podcast.
Cheers, Ben. Cheers, Mike.
Welcome everybody to the after party in the Pineal Room.
I could use some CMOS right now.
I could use some CMOS too. And Dr. Covell McDermott, shout out, has reemerged onto the social channels after a brief hiatus. I guess it was more than brief, wasn't it?
Yeah, it was a few months, maybe like all winter.
It was like all winter. Yeah, the man went into hibernation.
Yeah, good for him. We all should do that.
We should take the social media break. He's better for it, and he's back with a vengeance.
Yeah, but he's been hawking the CMOS, and it's making me desire his incredible blend. So I'm going to put an order in this week, I think.
Mm-hmm. And you sound like you might use a little CMOS.
I know. I got a little bit of an upgrade going this last week. Maybe it's the separation conflict from having to leave Ben in the North Country of my beloved New Hampshire.
I feel the same way. I felt really low, like real low energy the past week, really. And I was like, oh, I'm just depressed. That's what it is. And I'm not going to PorkFest every day. It's like, can we just make PorkFest a year-long celebration?
Seriously. It's so next level.
Life was so easy. It was so easy and straightforward. Go to PorkFest, hang out with people, listen to music, play music.
Yep. Share food.
Share awesome food. Go to
share
the river.
Ideas.
Mm-hmm.
Share camaraderie. Share rivers.
Yeah.
It's pretty freaking mellow.
Pretty mellow.
Yeah.
So you got a little upgrade, and okay. So separation conflict one. What else potentially?
It's a really good question, because June has been such a high water mark month, like just an epic month. PorkFest, Miles Music Camp before that. I mean, I was away from home for a couple weeks, but like that's my jam. That's how I roll. Having said that, apart from the delicious vittles coming out of your kitchen, I was at the mercy of other people providing food. The food at camp was really delicious and prepared with love, but I can't imagine it was locally, organically sourced ingredients. A lot of late nights, I gotta say, like 2 weeks solid of late nights, which is really not my routine. So maybe some sleep deprivation in there. And then the thing that just occurred to me today, although the Daniel Roydus observation about like differences in temperature and climate and humidity, and there's been like this sort of like wide arc of like super humid days, and then the storm will break, it'll get really cool and crisp, and I don't know, that could be contributing. But also, when I got home from being away at PorkFest for a week, and I hadn't seen the kids in a week, it's like, well, I gotta like do like a classic dad thing, like dad's home, and we just all went and got an ice cream, which is like not something we really do. And we just went, like, I live in New York State, and like strangely, one of the main purveyors of ice cream in New York State is the Stewart's gas station. I don't know if you know this. Like Stewart's has like that's one of their things. And I got something that I had put on Waylay for a few years was my annual banana split is what I'm driving towards. I hadn't had one of those in years, probably since we started this podcast. Because I had sworn off sugar for a long time, and I've sort of let it creep back in. And then, you talk to the Bigglesons, and they're like, "Eat your fucking ice cream, be a man, it's all good." But I did get sick right after that. Right after that. So I don't know, Ben.
This is what Dr. Tom Cowan says. You just let the person talk, and they will find.
They'll eventually arrive at the conclusion.
They will arrive at the conclusion. This would probably be a good time to tell you remember the beautiful like salads we were having, like breakfast salads.
Yeah,
lettuce that came right out of the garden.
Yeah,
I spray Roundup all over that. I spray. In fact, I have got like a specific like salt mixture that has little Roundup granules. So I just crack that right over the. That was just all over that meal.
It's artisanal Roundup.
Artisanal glyphosate.
I don't know. I mean, when I got home, I had such a good week. I was walking out of the reservoir. The weather was nice. I was swimming. I was taking in sunsets, and the other variable as I've been. I mean, maybe it's pointless to even itemize variables, but because this is what we do, it's kind of fun. Is I was doing a little weeding in the garden, and there was a bunch of this kind of beautiful wild mint growing all around one of my garden beds. And while I was sort of weeding it to just clear out the clutter from, the other, the things that I planted intentionally in that bed, and there was plenty of other mint all around the yard. I didn't need it there. But me being me, I was like, well, I don't want to let anything go to waste, even these quote unquote weeds. So I got my big glass, sort of carboy, server, and I filled it up with spring water. I did go to the spring as well. And I put all that mint in there, and I set it in the sun for like 2 days, or maybe a day. I don't know, 2 days, day or two. And it made this beautiful, like golden mint tea. But what I did not wash it. Like I should have washed it first. I just like got a little impetuous. I like harvested the mint. I threw it in the water. I didn't even clean it that well. Like there was some soil down in the bottom. And I was like, well, maybe that's part of like my impetuous nature. Like I could have been a little bit more careful with my mint tea. But anyway, that's all I got, buddy. I'm just throwing it all against the wall.
It doesn't sound like you to just pick something arbitrarily and then like eat it without preparation or consideration.
Not at all.
Very out of character. Very out of character.
Anyway, that's what I got.
Got it. Yeah. Well, hey, who knows? Point is, you're going to be better on the other side of this, right?
Yeah. Who fucking cares, really? I mean, I'm on the way. I just have sort of the residual clogged nose and throat right now. I do have a big hockey game tonight, so I got to rally.
You too.
And I was just on Jacob Diaz's Instagram, and he was saying he did a post about raw milk, and he did a raw milk cleanse, like 3 days of only drinking raw milk. So I'm definitely gonna do that, dude. That's next on my list. Maybe after hockey.
There you go. Speaking of mint, maybe what you need is just some more Dragon Slayer.
Oh yeah, the Ben and Grayson formula.
That's right. I did a little hike on Sunday, and came back, and I made an entire a colossal pitcher of Dragon Slayer just for myself. It was like a beautiful sunny day. Came back, like went to the river, came back, did some bunch of yard work, and I was like hungry, completely drained, and that's what I went with.
And so for the listener, that's watermelon.
Oh, right. Yeah. The Dragon Slayer is the drink that we made up while Mike was here. So my son Grayson and I. So it's watermelon, mint, like fresh mint from the garden, some freshly squeezed lime, maple syrup, and just a sprinkle of salt, and then just some ice and water, and you blend it up into this beautiful frozen drink.
Mm-hmm.
That's the summer of 2024. The official drink of the summer of 2024 is the Dragon Slayer.
That'll be in the Terrain Theory cookbook.
That's right. That's so called because it will slay the fire-breathing dragon. It is so cooling.
Yes, especially in peak summer.
Mm-hmm. Speaking of cool, Jamie Andrews and his project making science cool again.
Oh my God, citizen science! I was reminded of Mike Donio and how we were sort of playing with that idea, joking about like the crowdsourced science lab.
That's right.
And
Yeah.
and
He's doing it.
honestly, well, it sounds like he is investing in some equipment and moving towards a lab, but really it's having the vision, because you fundraise, and then there's plenty of labs that you can hire. They're just for hire, which I guess I didn't really think of it that way until this conversation. These are just like, they're engineers. You hire them to do a job and you give them the parameters and they spit out the data. So God bless them. Someone had to take the bull by the horns and do it. And it's just like I said during the interview, it was sort of one of those aha moments. Like, this is legit. This is what the internet's for. This is what they meant when it's like returning power to the people. This is the first time I've actually seen that play out to such a significant degree.
I wonder if there's the belief, I think I share, that the three-letter agencies and the industries themselves own the labs, and so they control all of it. But as Jamie's educating all of us on, they're actually for hire. You can find these labs that basically are for hire, and they will perform the experiment the same way that is done by the industry and the three-letter agencies.
Incredible.
Yeah, and I think, first of all, I would encourage the listener to head over to YouTube to watch, or at least go through the presentation, maybe at like 2x speed, so you can see with your own eyes the images under electron microscope, and the comparison between what the CDC says the measles looks like and SARS-CoV-2 looks like and HIV looks like, to what Jamie and his team, I would say actually not even Jamie, it's the lab that performed this, found in the control, basically the sample that could not possibly have contained a virus. And just see for yourself and draw your own conclusions, and also observe the cytopathic effect in their controls, and compare that to the cytopathic effect that occurs in these same isolation experiments that allege to contain the sample from a sick human, or the virus or bacteria, the contagion causing virus and bacteria.
Yeah, and I think it's also important to continue to point out that we're talking about cell lines from cancer patients from 60 years ago. Like, that in and of itself, how is that not a red flag? That has anything to do with a living, breathing human being right now.
Right. And virology, no one has ever witnessed a quote unquote virus entering a cell and performing all the functions that a virus is said to perform.
Yeah.
Hijacking the cell's mechanics and replicating and exploding and destroying it. None of that has ever been witnessed. This is the best that they have. And as Jamie's work and the team's work is proving, like, it's not science. It hasn't proved anything. Yeah, those unicorns, though. And what I appreciate about his work in this approach is that they took the criticisms that were heaped upon Stefan Lanka's work, and they've accounted for them, and they've gone with the most robust cell lining possible, so that no one can point at it and say, well, you picked the weakest one, and of course that's why it broke down and experienced CPE. And he's addressed and accounted for criticisms and said, now what do you have? Pointed it now. And also the pulling back of the curtain, I think, is a huge piece, that if you don't show the methodology of your controls, as is not done in virology, to me, Jamie says it's not fraud. I look at it and go, I think that's fraud. That's
Sure.
intentionally obfuscating or hiding what is the part about the experiment that makes it science. If you're not upfront about that, or you won't reveal that for whatever reason, that's not science any longer. It's no longer science.
Yeah, I like that he calls what he's doing Science 2.0. It's sort of like Tom Cowan's new biology.
Old new science. Yeah. Yeah. So folks, get over there. First of all, get to the video version of this if you get a chance, just to take a look at the slides and draw your own conclusion after seeing. Seeing is believing or can be helpful. And then please do consider heading to the link in the show notes to donate, because then you're invested. You're invested in this. And if you have any sort of question, whether one way or the other, if you fall, no, that this can't be real, viruses definitely do exist, well, fund the study, because the way that they're approaching it will, I think, is going to conclude one way or another fairly definitively.
Yeah, and as he says, a control is scientifically binding. So if you're in a position where you need some hard copy of the paperwork of those control studies to present in your defense, whether it's travel or school mandates or what have you, you're getting something for your money too, which is an incredible resource he's provided to the community.
Yeah. Shots were fired. I do know some folks who studied epidemiology, and when he said epidemiology is basically just storytelling, I felt like there were some shots fired there. But I guess when it comes down to it, that's really what it is, isn't it? They're not in the labs. They're just making observations and drawing conclusions based on correlation, not causation.
Right.
Yeah. Well, Michael, good luck in your game tonight. I guess by the time this thing airs, we'll know the result of it. This is a final, you said?
Yeah, it's the final for the spring league that I joined.
Okay.
Yeah, it should be fun.
And you're going to play forward for this one?
Yeah, they've got me in forward, which is fun. But I was saying to one of my teammates, for me, as a career defenseman, playing forward is like being on vacation. It's like you have far less responsibilities. Being a defenseman, like you got to like play defense. It's like your responsibility to like shut them down and keep the puck out of the net. Playing forward, that it's just a different set of responsibilities that they're completely foreign to me. It's like it's really stress free. You're supposed to score some goals, but if you're not expected to score a goal every shift, like you're supposed to get a couple goals. But it's someone else's job to keep the puck out of the net. Basically, you should help. Anyway, it's like being on vacation. I love it.
And a fruitful vacation too, right? You scoring hat tricks up there.
I did get a hattie last game. Yeah.
Right? The late night text with the image of you in the locker room with the, what is it, bird hat, the crow hat?
Yeah, the team is called the Ravens.
Ravens, the Raven hat, right? So that's what you score the hat trick.
Yeah.
Incredible.
No, I mean, I have to brag to somebody. Might as well be you.
Heck yeah, man. Heck yeah. Well, again, I high expectations on you, but none at all at the same time. Go have fun.
Thank you.
Hope your team wins, man.
Thank you.
Folks, nothing you heard here should be taken as medical advice, as neither Mike nor I are medical experts. Remember that you are light, you are love, unicorns don't cause sunburns, and you are your primary healthcare provider. Thanks for tuning in, and we'll catch you on the next one.